Autophagy is a tricky phenomenon to measure because there are very few labels which definitively label autophagosomes, so the vesicles which form and engulf cytoplasmic material and then deliver it to be degraded by the lysosome. So cells have you reuse multiple pathways to make vesicles and traffic them around. And there is one sort of unique label for autophagosomes. It's called LC3. LC3 exists in the cytoplasm of cells, but the unique form of it, which labels autophagosomes, is a lipidated form. It's called LC3-2. So using a fluorescent tag on that protein, we can see autophagosomes in vivo. So we can see the fluorescently labeled small vesicles, puncta, as they're referred to, labeled with this LC3 tag. Now, the only reason we can tell the difference between LC3 in the cytoplasm and this LC3-2 form, which is the unique label on the autophagosome, is that we see the fluorescence become bright in these puncta as they cluster around the autophagosomes. So we use that as our best label for seeing autophagosomes in vivo, and we can count their number in cells. We can see how they're trafficked and look at their turnover. So we have this one marker, and we can label it with fluorescent proteins. And the real sort of leap that we've made recently is to change the nature of that fluorescent tag so that we can better follow these autophagosomes. So we have dual-colored fluorescent proteins. So we can see we can use them to discriminate the pH within the cell. So in normal neutral pH conditions, we would see a green and a red fluorescent reporter. Whereas, for example, using one of these fluorescent probes where the fluorophore is sensitive to pH, then when this autophagosome fuses with the lysosome, you lose the GFP signal. It's quenched in the acidic environment. And so you can measure the colors of these autophagic puncta, LC3 puncta, and count how many autophagosomes you've got and whether they're making it to the lysosome to be successful. So there's a range of these in vivo probes. They can be two-color fluorescence, or they can be probes where the fluorescence emits at a different wavelength, depending on whether it's in a neutral or acidic environment, so in the cytoplasm or in the lysosome. Those are great tools, but the challenge is, are we really wanting to study just counting autophagosomes and where they might be, or do we want to couple that with the ability to actually measure the substrate as well? So should we be measuring the autophagosomes, or should we be measuring the autophagic substrate, the aggregate-prone proteins that we think are the culprits in causing these diseases?